ahead.
Thank you. Thank you for the introduction. Good morning, everyone. First, I want to thank the organizers for giving me the opportunity to present our work. The title of my presentation today is Reunion, Transcription Factor Bending Prediction and Regulatory Association Inference from Single-Cell Multi-Omics Data. Single-cell multi-omic technology can simultaneously profile gene expression and chromatin accessibility from the same cell, which provides the opportunity to integrate different data modalities to investigate the gene regulation mechanisms underlying cell fate decisions. Transcription factors, regulatory sequence elements within accessible genomic regions, and target genes are key components of the gene regulatory networks. We want to decipher how transcription factors or TFs regulate target gene expression through interaction with the cis-regulatory regions. The region TF gene triplet associations are highly informative for understanding the gene regulation mechanism. However, there are computational challenges in inferring the regulatory associations due to the combinatorial complexity of gene regulation and also the noise and sparsity of the data. The two major challenges we want to address in this work are how to identify triplet regulatory associations by more efficiently utilizing different information and overcome the limitations of the multi-scanning approaches which were widely used in their bending prediction. Here, we develop a computational framework reunion which performs genome-wide bending prediction and regulatory association inference from single-cell multi-omics data. Reunion incorporates two methods, unify and rediscover. Unify aims at inferring triplet regulatory associations by integrating information from different components simultaneously. Rediscover is focused on tier bending prediction without the constraints from multi-scanning approaches. More specifically, the current methods in single-cell multi-omic data analysis mostly only utilize information from two components to estimate the pairwise associations between region TF and gene separately, as pairwise associations may also depend on the third variable. Using only two components may lose critical information. Two recent methods, Pendulum and Tripod, utilize three components to estimate the triplet associations. However, both methods have high time complexity and limitations in mechanisms that may miss regulatory associations. Moreover, the current methods all rely on TF bending motif scanning to establish the association between region and TF. In motif scanning, we use a collection of non-sequence patterns of TF bending sites, which are motifs, to scan the DNA sequences of accessible genomic regions and identify potential TF bending sites. Here, we use the ataxic peak loci to represent the accessible genomic regions. In the motif scanning results, we may have correct detection, wrong detection, and missing detection or false negative. The missing detections may be caused by incompleteness of motif collections, limitations of multi-scanning algorithms, and typical bending behaviors. The current dry inference methods cannot recover the missing detections. If there are no motifs detected for a TF inner peak, the peak is not linked to the TF. The missing peak TF links may cause missing TF gene regulatory associations and hinder the discovery of potential combinatorial TF regulation. There are also supervised learning methods which predict TF bending by utilizing DNA sequence features in combination with epigenomic data, which are mostly back sequencing data. These methods don't require multi-scanning, but they require chip-seq data for model training, which are costly to obtain and only available for few TFs. With these challenges in consideration, we developed our framework Reunion, which integrates two cooperative methods, Unify and Rediscover. Unify utilizes complementary information theory inspired score functions to take into account peak, TF, and target genes simultaneously to infer the triplet regulatory associations from single-cell multi-omics data. Rediscover takes peak TF links predicted by Unify as input, performing pseudo-semi-supervised learning to predict TF binding in genome-wide peaks with or without the motif detected for a given TF. Our first method unifier has three components, including peak gene assignment, motif scanning, and the core module of peak TF gene triplet scoring. In this module, we integrate two score functions to quantify the strength of a triplet link. Score function 1 estimates the shared information between TF and the putative pair of peak and target gene, aiming at predicting TF binding by not only utilizing peak TF correlation but also incorporating information from the potential target gene of the peak. We want to estimate the joint mutual information between the TF and the peak gene pair. The joint mutual information, or JMI, is the sum of the mutual information between the TF expression and the peak accessibility and the conditional mutual information between gene and TF expression given the peak accessibility. In practice, it is challenging to estimate joint information for continuous variables from the limited data with sparsity and noise. Instead, we use correlation to approximate the mutual information and use the partial correlation between gene and TF expression, even peak accessibility, to approximate the conditional mutual information. Score function 1 assumes that the target gene of a peak is given, however, there is uncertainty about the peak gene associations. We then define score function 2, which estimates the shared information between a peak TF pair and the target gene. Assuming TF is bounding to the peak, we want to estimate the joint mutual information between the peak TF pair and the potential target gene. Again, we use correlation and partial correlation to approximate the mutual information and the conditional mutual information separately. We also utilize a regularization term which takes into account the motif scanning scores and peak accessibility into consideration. The two score functions are complementary to each other and jointly quantify the association strength of a peak TF gene triplet link. We then use thresholds on the two scores by unified, and we reduce the triplet links to peak TF pairs to give as input to rediscover. RegSys utilizes a pseudo-semi-supervised learning approach to predict TF binding in genome-wide peaks for a given TF. In semi-supervised learning, we have a small amount of labeled data and a large amount of unlabeled data. We want to utilize both groups' data to learn prediction models. In our case, the labeled data are peaks with non-binding status for a given TF. The unlabeled data are peaks without the non-TF binding status. In reality, we only have unlabeled data as we don't know the ground truth of TF binding. Our approach is to select the pseudo-labeled training samples based on the peak TF links predicted by Unify. Here, we have the new challenge of how to select the training samples. Then we have RegDiscover. RegDiscover has multiple connector modules including feature representation of peaks, peak clustering, peak enrichment analysis, pseudo-labeled training sample selection, and prediction model training. First, we assume that peaks bound by a specific TF may show distinctive chromatin accessibility and sequence patterns that can discriminate them from unbound peaks. Here, we have a peak accessibility matrix, which is the accessibility of each peak in each metacell. We also have a peak motif matrix, which is the binary presence of TF binding motifs in each peak based on motif scanning results. We perform singular value decomposition to factorize the peak accessibility and peak motif matrices to obtain low-dimensional embeddings of the peaks. With the chromatin accessibility and sequence feature embeddings, each peak is placed into two feature spaces. We then perform clustering of the peaks in the two feature spaces. We use the phenograph method for clustering. Each peak then has a paired group assignment, which corresponds to the pair of its cluster labels in the two feature spaces. We also identify the k-nearest neighbors of each peak in each feature space. We assume that peaks with similarity in both feature spaces are more likely to show the TF binding status. Next, we perform enrichment analysis of the peaks with TF binding predicted by unifier in the paired groups. We compute the number of genome-wide peaks in each paired group to estimate the background distribution, and compute the number of peaks with TF binding predicted by unifier for the given TF. We then perform Fisher's exact test to estimate statistic significance that peaks with TF binding predicted by unifier are enriched in a particular paired group. We assume that peaks from the paired group enriched for TF binding peaks predicted by unifier are more likely to be bound by the corresponding TF. Then, we perform pseudo-label training sample selection and prediction model training using the paired group assignment and the peak enrichment significance as guide. We select the pseudo-positive samples from the peaks with TF binding predicted by unifier. We use adaptive thresholds on score 1 by unifier and also the peak accessibility TF expression correlation depending on whether peaks are from a paired group enriched for the TF binding peaks predicted by unifier. We select the pseudo-negative samples mainly using the criteria that they are not similar to the unifier predicted TF binding peaks based on the paired group assignment and the neighborhood relationship in the feature spaces. We concatenate the commutative accessibility and sequence feature embeddings to obtain a feature vector for each peak. We then train a classification model using logistic regression and apply the trend model to genome-wide peaks to predict TF binding for the given TF. We apply the reunion to single-cell multi-omics human PPMC data from 10S genomics to infer triplet associations and perform genome-wide TF binding prediction. We evaluate TF binding prediction performance using TF ChIP-seq datasets downloaded from public databases. We compared with 3 categories of methods, including GRN inference methods utilizing single-cell multi-omics data, TF motif scanning-based prediction utilizing different motif collections, and TF footprint analysis-based prediction, which is Tobias. For methods that provide continuous peak TF association scores, we evaluate using AUPR. There are also methods providing binary predictions without scores. Both methods can be evaluated using inference score based on the binary predictions. We found that reunion outperformed other methods on average performance in AUPR and inference score. In the example plots, we show the pairwise comparisons using AUPR and inference score for each TF ChIP-seq dataset between reunion and the representative compared methods. We found that for the majority of the TFs, reunion can retrieve performance improvement compared to the other methods. We also evaluate the prediction performance using precision and the specific recall. Reunion had similar performance to Tobias at precision and 50% recall, and higher performance at precision at 20% or a higher recall. Next, we use streaming to perform sequence motifs discovery in the peaks without motif detected, but with TF binding correctly predicted by rediscover based on the evaluation using TF ChIP-seq data. One of the reasons that motif scanning may not identify motifs in a peak is that some non-canonical motif variants may not be well represented in the motif collections. For example, Sujemi identified potential dimer motifs of GATA3, which are different from the motif in the curated CSBP motifs collection we used for motif scanning in reunion, and also deviate from the other non-GATA3 dimer motifs in the motif databases. Furthermore, for each TF with TF binding predicted by rediscover in the peaks without the motif detected, we link the TF to the potential targeted gene through the candidate peak gene links previously identified by unifier. The new predicted TF links establish new peak TF gene links. Here, we focus on the scenario where new gene TF associations may be generated. we perform gene expression prediction using the TF expression as predictors, and we compare the prediction performance between utilizing two groups of predictors. Both groups contain the TF regulator previously identified by Unify for the target gene, and the second group additionally includes the newly associated TF. We observe that for many TFs, there may exist potential new target genes for which including this new TF as a new predictor may improve the gene expression prediction accuracy. Here is in the example plots, each data point corresponds to a potential new target gene of the given TF. The observations suggest that performing TF banding in peaks without motif detected may help recover potential missing TF gene regulatory associations. In summary, we develop a reunion and integrated framework for genome-wide TF banding prediction and regulatory association inference from single-cell multi-omics data. Reunion integrates two cooperative methods, Unify and Rediscover. Applications to the human PBM's data show the performance improvement of our method compared to other methods in TF banding prediction, and also the potential of our method in helping to discover the potential new TF gene regulatory associations and TF banding sequence patterns. In the acknowledgement, I want to thank Dr. Dana Pell, Dr. Taunoy, Dr. Cassandra Baziak, many other lab members, and Dr. Manu Sethi for the helpful discussions. Thank the funding resources. Thank you very much for your attention.
Thank you, Yang Yang. We have time for one small question, if there's anyone in the room. Yeah, so now, go Shmita. You just have to, shout a lot.
So I actually had two questions, but maybe
No, just
closer.
Yeah.
There. Okay,
Very close.
okay. So I think so. Your so nice work, by the way. So your pipeline seems to have a lot of different steps, and I was wondering how sensitive are the results to each of the different steps. For example, the pseudo bulking, like instead of using C cells, what about using a different method? How do you control the resolution? And then for clustering, how do you know which method? And so do you have a sense of that?
Yes, thank you very much for the question. Yes, so actually, as our method has multiple steps, there are many parameters to control. We have did some experiments on how the varied parameters may affect the performance, and some performance are quite consistent, but we haven't performed the testing of all the parameters. Yeah, this is the steps we'll be working on.